Okay, wait a minute. Actually, I forgot something. Okay. Fine. So this all we have discussed yesterday only, and then we have discussed another important mechanism, that is uh, chymotrypsin mechanism actually. So this chymotrypsin is having the same mechanism, which is also shown by several enzymes in this category. So this family is made comprising a big family, and all the enzymes are called serine proteases because they are having a very sensitive serine residue or their active site. So this serine residue is catalyzing the most of the reactions. Along with serine, there are two other important amino acid residues in these proteins or enzymes, which are responsible for catalyzing the reaction. So this is called catalytic triad. So this catalytic triad is present in trypsin, along with chymotrypsin, elastase, thrombin, thrombin enzyme, subtilisin, acetylcholine esterase. So all these enzymes, and there are other related enzymes also. If you do not know the names, actually it is given in your reference book. So if you move a few pages, you will get a table where they have shown each enzyme. Most of the enzymes which follow this serine protease. So these are serine proteases. This is a complete table. So it includes trypsin, chymotrypsin, elastase, thrombin, and these are not shown in your pathfinder. So plasmin, it is an enzyme which is responsible for dissolution of blood clot. So once the blood clot is formed by clotting mechanism, so there is a protein or the enzyme plasmin which is responsible for the dissolution or the removal of blood clots. So it is very important enzyme to remove all the clots from the circulation. Then there is the enzyme calcarein, other acrosomal protease. It is present over the sperm head so that it is able to penetrate into the ovum or egg. There is lysosomal proteases which are present in the suicidal bags of the cell. These are called lysosomes, and these are responsible for removal of certain proteins which are not necessary for the cell itself. Alpha lytic proteases, subtilisin, yes, we have discussed this, proteases A and B. So these are all important enzymes for somewhere in digestion. Okay, so in our digestion process, they're all having their roles. So now the question is how they are able to catalyze the reaction. So you should know these are all serine proteases. So more or less they are having the same mechanism. So mechanism is very simple again. So there are 3 amino acids this time over the active site of the enzymes. So these are having aspartate, histidine, and serine. So this serine is very important. Therefore, they are called serine proteases. And this is called catalytic triad because all the 3 amino acids are in close proximity and they are having hydrogen bonds in between them. Therefore, they are making some kind of proton channel. Okay, so they're bound by hydrogen bonds. So this is a hydrogen bond. The dashed lines are hydrogen bonds. So the serine, histidine, and aspartic acid or aspartate, these 3 amino acids are very important in serine proteases. Then we have discussed RNase A also. RNase A is having 2 amino acids, two acidic amino acids. Both are histidine. One histidine is present over the 12th number position. Second is present over the 119 number position. So they are involved in acid-base hydrolysis, acid-base catalysis, sorry. Catalysis using these 2 amino acid imidazole ring. So this ring in the histidine is called imidazole ring, imidazolium ion. Okay, so they help in catalyzing the reaction, and because RNase A is the most studied example, so therefore this RNase A is responsible for the hydrolysis of RNA. And you know the mechanism, because the RNA is having ribose sugar instead of deoxyribose. So this second prime carbon is having OH group instead of H in DNA. So H is present in DNA, but this is RNA, so obviously it will have an OH group. So this OH group first will be functionalized with the help of this lone pair of electron. So this hydrogen will be accepted by this particular histidine, which is present in the RNase enzyme. So because this is a protein, RNase protein, So this will have a chain of amino acids. So two important amino acids in this chain are these two histidine over these two positions, 12 number position from the N terminus. Suppose this is the N terminus, this is the C terminus. So the first will act like a base because it will accept this particular proton. So you know bases are acceptors of protons, and whereas donors of protons are called acids. So first step involves this is a base, this is an acid. And the second step, this will act like an acid, this will act like a base. This is called acid-base catalysis. That's why it will able to hydrolyze the RNA. Now, the last enzyme in this category is called aspartate proteases. These aspartate proteases are okay. So these are having some aspartic amino acid over their active sites. So beside the serine proteases, which we have discussed in case of, and there are several different examples we have discussed. So these include trypsin, chymotrypsin, elastase, thrombin, plasmin, calcarein, acrosomal proteases, lysosomal proteases. In some cases, in the complement system, C1 actually, this is a part of immunology though, but if you can remember, this is also important. Then there is some proteases A and B and subtilisin. Okay, so these are all examples of serine proteases. But because we are going to discuss one another category that are called aspartate proteases, so they will involve two more important enzymes, and you need to remember the names of both these enzymes because they are very important. One is called renin. So if you can find something, renin. Renin is involved in the blood pressure regulation. If you remember the renin, angiotensin, aldosterone system. So, okay. So first of all, we will read this text. Then I will tell you. So one is renin, which is showing aspartate protease mechanism of catalysis. Second is pepsin. So if you remember pepsin is an enzyme which is able to cleave uh, antibodies at a specific position. So this is also a part of immunology, but I'm giving you a hint here. So this pepsin is actually cleaving the antibodies as at a specific site so that the FC fragments are lost. Okay, so I will discuss this. If you want to know more about the pepsin mechanism of hydrolysis of antibodies, you can go to page number 517 in your Pathfinder part first. So page number 517 of first part of your Pathfinder. Actually, I'm having 7th edition, so I'm exactly telling you the page number 517. So there they have shown how the antibodies can be hydrolyzed using two enzymes. One is called papain, second is called pepsin. So this is the one application of this pepsin. Now we will look how this pepsin is produced, and actually it is produced by the chief cells of our pancreas, and then it is released into the stomach. Okay. So besides serine proteases, we have discussed so many chymotrypsin, trypsin, elastase, etc. Other class of proteolytic enzymes utilize either cysteine, that are called cysteine proteases, and actually we are more interested in aspartate proteases because they are very important enzymes again. So aspartic acid, if it is catalyzing a reaction through aspartate, then it's called aspartate proteases to perform their catalytic functions. And aspartate proteases, it is actually the two aspartate residues or amino acids that make the catalytic machinery of that particular enzyme. So the carboxylate group of the two aspartic acid residues, if you remember the aspartic acid, so aspartate will have a CH2COOHR group. Okay, so it will assist in proton transfer because it will have COO negative or can be converted into COOH. Therefore, it will assist in proton transfers from a water molecule. Now, who is going to donate the proton? The water molecule here. So this is important. Water molecule is going to donate the proton this time and act as a nucleophile, so that this nucleophile, because it will have a negative charge, OH negative, so it will act like nucleophile, which will attack over the peptide bond of the peptide or the protein and will cleave this particular peptide bond. Okay, 1 minute. Fine. So we were discussing Okay, so aspartate proteases are found in all forms of life. They're present everywhere, so they are ubiquitous. The proper term is ubiquitous if anything is found everywhere in the universe. Members of this class include renin, enzyme having role in regulation of blood pressure, and then there is one digestive enzyme called pepsin. Okay, so these two enzymes are very important. One is called renin, and one is pepsin. Then there is one more enzyme. The third enzyme in this class, aspartate protease, is HIV protease. This particular enzyme is very important for the virus itself. So HIV virus is human immunodeficiency virus, which is the causative agent of AIDS, acquired immunodeficiency syndrome. So this is also a member of aspartate protease family of proteolytic enzymes. HIV protease plays a very critical role in the life cycle of the virus, and for this reason has become an important target of the drug development. Okay, if you now why this class is important, if you know the mechanism of aspartate proteases, you are able to design some important drugs. Okay, so the drug development in the battle against the HIV infection. So therefore, these mechanisms are important to consider. Now there are two enzymes we have discussed. One is renin, one is pepsin. So let's look into the roles or the applications of these two enzymes so that you can remember. Oh, okay. So if you will not able to understand their applications or roles, then somewhere or somehow you will forget them. So we will discuss some of the applications. So first of all, this is called renin angiotensin aldosterone system. So there are three words. So renin is an enzyme. It is an aspartate protease. Angiotensin, mechanism of regulation of blood pressure, and the one important uh, aldosterone. Okay. So renin is produced by the kidneys. Uh, okay, and control the activation of the hormone angiotensin. So angiotensin is an hormone which stimulates the adrenal gland to produce another hormone that is called aldosterone. So this is an enzyme. This is a kind of aspartate protease or enzyme which is produced in the kidneys. Then it will activate, it will control the activation of this hormone angiotensin, and this in turn regulate or stimulate the adrenal glands. These glands are present over kidneys. So adrenal gland produce an aldosterone. So finally, this aldosterone. Okay, so produced in response to renin. When this renin is produced by kidneys, so kidney produce this particular enzyme renin when there is a drop in the blood pressure. So if there is a low blood pressure, so your kidneys will start releasing the renin enzyme, so that finally the angiotensin and the aldosterone will be produced. Or a decrease in sodium chloride concentration in the tubules of the kidney. So there are two conditions in which your kidney can release this particular enzyme. If there is a decreased blood pressure or drop in blood pressure, or there is a less concentration of sodium chloride ion concentration in the tubules of your kidneys. Okay, so then there is an important diagram also from where you can get the image of this. So suppose there is dehydration. So when you will have a low blood pressure, suppose you are undergoing dehydration, you are taking less water, or there is a deficiency of sodium chloride ions, or there are some problem, hemorrhage. So all these conditions. Okay, so finally, what is the result? Aldosterone will be produced as this regulation is happening because renin is inducing the angiotensin. Angiotensin is inducing, stimulating your aldosterone. Aldosterone is produced by the adrenal glands. So help in regulation of blood volume, pressure, as well as the levels of sodium, potassium, and H positive ions in the blood so that the blood pressure can be maintained. And in short, this is called RAA pathway. So there is a decrease in blood volume. If there is decrease in blood volume, so decrease in blood pressure also. Finally, your kidney. It will stimulate your kidney. So this is juxtamedullary cells of the kidneys, which is going to produce a renin hormone. Once this renin hormone will be produced, it will stimulate along with from the liver. Actually, it is coming angiotensinogen, which will be converted into angiotensin 1 and then angiotensin 2 with the help of some enzyme which is coming from the lungs. This is called ACE, angiotensin converting hormone. It will convert the angiotensin 1 into angiotensin 2. Finally, this angiotensin 2 is the active form, so it will produce two type of responses. One, it is acting over adrenal cortex cells so that it can release or stimulate the aldosterone production. Also, it will work like vasoconstriction of arterioles. So what is vasoconstriction? To constrict. Okay, so that blood pressure is increased until it returns to the normal. Finally, if it is working over adrenal cortex, something that is increased K positive ion concentration and extracellular fluid also working over adrenal cortex so that more and more aldosterone is produced. So it will lead to the increase in positive and water resorption in the tubule so that the blood volume will be increasing and again it will come to normal. So this is a mechanism of blood pressure regulation and blood volume regulation also. Second thing, so this is about renin. Second, we have discussed about the pepsin actually. So pepsin is another, so where is pepsin exactly? 1 minute. Okay, so here. So pepsin is actually working as like this so that it will cleave the antibody over the Hinge region. You know Hinge region is a part of antibody where it is attached via two disulfide bridges. So these two disulfide bonds are remain as such, and the remaining portion below this antibody, actually there is another part that is called Fc fragment. If you do not remember the structure, let me draw it. So suppose it is having two Y chain, it is a kind of Y chain or the Y structure shape of antibody. These are having two light chains. Okay, let me draw with a different color. So green color are called heavy chains, red colors are called light chains of antibody. And you know there are certain disulfide bonds. So let me draw with the yellow color. So there is a two peptide bonds in this Hinge region. So this is called Hinge region. And there are other disulfide bonds which are connecting your light and heavy chains. Now this portion is called FAB segment, fragment of antibody, FAB fragment. So this is called FAB. Why this is called because this will going to bind with your antigens. So here it is having some sites where your antigens will come. Let's draw like antigens like this. So these are antigens. Suppose they are going to bind with this FAB fragment. And there is another section. This is called FC fragment. So this is actually your FC fragment. So actually I made something very. So simply this is called FC fragment, fragment of crystallization. So what will happen actually when this pepsin is going to break your antibody, it will cleave your antibody somewhere here so that suppose if it is broken here, so finally these FC fragments should be remain intact, but what will happen? They will remain intact in case of papain hydrolysis. But here, because this pepsin is having some non-specific cleavage, so it will cleave these so that there is no FC fragment is recovered. So the smaller fragments will be lost. So we will recover only this fragment. This is a big fragment. So this is called FAB double dash, FAB2 double dash, FAB2 dash. So this fragment will be recovered. So this is, and these are lost, FC fragments are lost. So this is given. Okay. Hope this is clear. And if you are not able to understand, you can get from page number 517 in your book. Okay, now I will come back to, so we were here actually, okay. Now, okay, so some noble terms actually left which we are going to cover in this class only. So these are also very important. So have you heard about some terms, for example, Abzymes, ribozymes? Okay, so these terms are also very important. So somewhere a direct question can be asked or in some other form. But before starting that part, actually, I'm left with this some text in your book. So actually, we have discussed all the three major catalysis mechanisms. So these are catalytic strategies. We have discussed covalent catalysis. We have discussed acid-base catalysis. Also, we have discussed metal ion catalysis using various kinds of examples. And I hope you are able to get at least something out of this. And these all three are very important, and the most important part is some of the enzymes are employing more than one catalytic strategy to catalyze a reaction. For example, we have seen in case of lysozyme. Beside this, we have also discussed some other things, for example, proximity and orientation effect of enzyme, or some other type of rotation of, or the strain mechanisms. Okay, so what is proximity and orientation? Proximity and orientation is a hypothesis in which an enzyme uh, enable the substrate so that it is holding the substrate in such an orientation so that it can interact more efficiently, and it will finally result into the product formation. Okay, so it will just increasing the binding energies of some of the bonds so that the transition state energy can be decreased and more number of substrate can be converted into the product. Whenever we are talking about a chemical reaction or a biochemical or a catalytic reaction, simply, we should express the activity in some units. So these are of two, there are two kinds of units. One is called as enzyme unit U. So this is the representation. If you find U somewhere, it is simply the representation of enzyme unit. So you should understand now what is enzyme unit, what is cattle. Okay. So first of all, the amount of enzyme can either be expressed as molar amounts or measured in terms of activity. How you are able to know how much enzyme you are having in your hand? So obviously, you will take the enzyme in a test tube, you will add some substrate, and you will catalyze a reaction, and finally you will measure the enzyme in terms of its activity, how much of the substrate it is able to convert into the product. So the enzymes are usually present in very small quantities, because you know the enzymes are present in small quantities. Therefore, they must be given some of the units which are showing some sensitive measurements. Okay, so for example, if an enzyme is able to convert 1 micromole of substrate into the product in 1 minute, then this unit is called enzyme unit. So 1 micromole is obviously very less, and we also know that enzymes are generally present in less amounts. So suppose we are having 1 micromole of an enzyme in our test tube. So how we know this is 1 micromole or 1 unit? So if this particular enzyme is able to catalyze a reaction, it will able to convert 1 micromole of substrate into the product in 1 minute. Then only we can say the enzyme is having catalytic activity of 1 unit. Okay. So a convenient method of enzyme quantification, if you want to know the amount of enzyme quantification, or you quantify the enzyme, so you need to measure the catalytic activity. Activity is then related to the concentration somehow. More of the concentration of enzyme you have in your test tube, the more of the substrate you are able to convert into the product. When the enzymes are quantified related to their activity rather than the direct measurement of the concentration, the unit used to report the enzyme level or activity unit. So these are called activity units. So you need to know the names of two activity units here. So the two standard units which are used in enzymology, one is called enzyme unit. Enzyme unit is the amount of enzyme or the concentration of enzyme causing the transformation or conversion of 1 micromole. 1 micromole is very small. It is less than 1 mole. So it is actually 10 to power minus 6 mole. Okay, so let me write it. Okay, so this is 10 to power minus 6 mole. So this is very small than a 1 mole. Okay, so if this substrate per minute, and this time point is also very important, per minute at a specific temperature, room temperature 25 degrees Celsius, under optimal conditions of measurement. If everything is fine, then if an enzyme is able to convert 1 micromole of substrate per minute at 25 degrees Celsius, then it is called 1 enzyme unit. So it is represented like 1 micromole converted per minute. Okay, the second unit is cattle. Cattle is actually the big unit, and therefore cattle is the SI unit of enzyme activity. So one cattle is the amount of enzyme. Here you will take 1 micromole, but here in cattle, the amount of enzyme which will able to catalyze the transformation or the conversion of 1 mole of a substrate. So here you are taking 1 micromole, but here you are taking directly 1 mole of substrate. And suppose there are second thing that is per second. So actually, first thing, you are having more substrate in your hand. Okay, so 1 mole of substrate, and you are looking that the enzyme which is able to convert the into product per second. Here you're looking for 1 minute actually. So this is also the 60th part, because 1 minute is having 60 seconds. So you are able to solve some of the very simple question which can be designed based on this. Here you are having less substrate, you are converting it per minute, but here you are having more substrate converting in less time, 1 per second. So obviously, if someone asks you some simple relation between a cattle and a unit, so the 1 unit is equal to 16.67 nano cattle, which is actually derived from here and here. So 1 cattle is 1 mole per second. So 1 mole of the substrate converted into the product in 1 second actually. So this much of reaction can be shown by 1 cattle of enzyme. So if someone asks you which is the big unit, obviously cattle is a big unit, enzyme unit is very small unit when compared to this, and the relation between the 2 unit, 1 unit is only the nano cattle. Nano is 10 to power minus 9. So this is very small unit when compared to a cattle. So okay. Second thing is, in today's class, the important points are some terms we will learn in this class. So hope this will not. Okay, so before this, actually, we also have one more page. Actually, in this PDF, when I made the PDF, I changed the number actually based upon your, so that you will not face any problem based on your convenience. Actually, I changed the page number. Otherwise, everything is fine. All the things are given in your Pathfinder only. So first of all, we will learn about isozymes. So if you know isozymes are having same function. Okay, so they are, because they are going to catalyze the same chemical reaction. So isozymes are called isozymes or isoenzymes, whatever you call, are the enzymes that differ in amino acid sequence. So they may vary in some amino acid sequences, but they are found to catalyze the same reactions. Therefore, they are having the same function. Now there is another term, allozymes. Okay, so suppose isozymes. So isozymes or isoenzymes, they are encoded by different genes located at different different spaces or loci. Actually, loci are called the exact points present over the chromosomes. So they are encoded by different genes which are located at different loci over the chromosome. But these allozymes are enzymes that arise from the same gene. Okay. So isozymes are enzymes usually display a different kinetic parameter or different regulatory properties, whereas these isozymes differ from allozymes, which are the enzymes that arises from the alternative form of the same gene. Have you heard about alleles? Alleles are the alternative forms of the same gene. And if these allele encode to different enzymes, then these allele encoded enzymes are said to be allozymes. Okay, because they are produced from alleles. A common example of isozyme is lactate dehydrogenase. So actually this table is showing the lactate dehydrogenases, 5 different type of lactate dehydrogenases. These are examples of isozymes. Okay, So these are present because these are found in different tissues. So for example, lactate dehydrogenase 1, 2, 3, 4, 5. There are 5 major types of lactate dehydrogenase. One is present in your heart and RBCs. Second is present in reticuloendothelial system. Third is present in lungs, then kidneys and placenta, and 5 is present in liver and striated muscles of your skeletal system. So because they are made up of different, they may be either homomers or hetero tetramer. Homo tetramer is made up of same subunits, so H4, HHHH, and this is M. So actually this is present in heart and this is present in muscles. Therefore, they are said to be H and M. So H is coming from heart, this is coming from muscle. So majorly there are two major variations, two different kinds of lactate dehydrogenases. So if all the subunits are same, then they are called H4. So these are called homo tetra. Okay, so somewhere it is written actually. So what is the role of this lactate dehydrogenase? You know, when you do some physical exercise or physical activity, suppose you are going for a run or gym and you did lots of physical exercise in one day, then there is a muscle pain in your body. Okay, so your body will not move the next day. This is because there is lots of lactic acid which is accumulated in your muscles, which is resulting in your muscle pain. Okay, so what will happen actually, this pyruvate is the end product of your glycolytic pathway, and when you do physical exercise you need more and more energy. So most of your glucose will be broken down into the pyruvate, because you will get most of your energy during such exercises from the glycolytic pathway, and it will result into lots of pyruvate. Then this lactic acid, which is present in your striated muscle cells and skeletal muscles, it will convert this into the lactic acid or lactate, and this will create the problem actually. So the reaction which is catalyzed by lactate dehydrogenase is conversion of pyruvate into lactic acid. Human beings have two isozymic polypeptide chains for this enzyme. One is called H and one is called M. H is present in heart, as I told you, M is present in muscles. So this is made up of two isozymes. One is H and M. Now, the different types of further classification means lactate dehydrogenase 1, 2, 3, 4, 5. They are made up of different combinations of these two isozymes. Functional enzyme is a tetramer. So the tetramer can be a homo, a tetramer, or heterotetramer. Okay, so the two different subunits combine randomly with each other to form the 5 isozymes, 1 to 5. And the kinetic properties of various lactate dehydrogenase isoenzymes differ in the terms of their relative affinities, various substrates, and their sensitivity to the inhibition by the product. So what is the difference between these? So they are having different affinity for the pyruvate, and they are having different inhibition kinetics using this lactate or lactic acid, which is produced as a result of this enzyme. Okay. Similarly, there is a second example given in your textbook that is called hexokinase. We have also discussed hexokinase 2.7.1.1 and last two. So these two are hexokinase, but actually there are 4 types of hexokinases from 1 to 4. Or somewhere you will find A, B, C, D. So they are only showing the 4 different types of hexokinases possible. So first of all, the function. So if you know the hexokinase, it is a kinase overall. So hexose is a sugar, either a glucose or mannose. And kinase is an enzyme which is transferring the gamma phosphate from the ATP over this hexose sugar. Now the first, second, and third hexokinase catalyzes the formation and the polymer, this phosphorylation of hexose, for example, glucose or mannose. The 3, 1, 2, and third, these 3 hexokinases are capable of phosphorylating several hexoses, whereas the 4th one is very specific, either hexokinase D or hexokinase 4. This is termed as glucokinase because this will specifically phosphorylating the glucose, okay, act only over the glucose as a hexose. So this one is very important. Hexokinase 4 is actually called glucokinase. So you should remember this line actually. Glucokinase is mainly expressed in the liver and pancreatic beta cells. So where you need the glucokinase, obviously where there is more of glucose. So pancreatic beta cell and liver, because in liver the glucose is going to convert into the glycogen. So therefore the glucokinase is specifically especially expressed in these type of cells, and characterized by the low affinities for the glucose substrate, and it is having less affinity for glucose. Now remember, these three hexokinases, 1, 2 and 3rd, they are having high affinity for their substrates. Suppose and remember, whenever there is high affinity, it will also have some consequences. देखो अगर किसी चीज का फायदा है तो उसका नुकसान भी होगा. Obviously they will be inhibited by their product. So what is the product? This hexokinase will convert the, for example, glucokinase will convert the glucose into glucose 6 phosphate, and then if they are having more affinity for the this product or this substrate, finally this glucose 6 phosphate or any hexo 6 phosphate will be result in the inhibition of that particular enzyme by feedback mechanism. So therefore this is not a good thing for the enzyme. Therefore hexokinase 1, 2, 3 are referred to as low Km, because what is Km? Km is that particular substrate concentration at which the reaction velocity is half V maximum. Suppose if an enzyme, I'm talking about all the three, 1, 2 and 3rd hexokinases, they are having low Km, therefore they are very sensitive for hexose. If they are sensing very low concentration of hexose, they will be activated, and therefore they will convert that hexose into the phosphorylated form. So this low Km isoenzymes, because of the high affinity for glucose, even at a low concentration, because glucose is also a hexose, so therefore if they detect very low concentration of glucose, they are going to phosphorylate this one. Obviously they can sense below 1 millimolar. Okay, all the three are strongly inhibited by their products, so this is a bad thing actually. So once they are going to phosphorylate the glucose 6 phosphate will be formed, and because they are having high affinity, it will going to inhibit that particular enzyme, either 1, 2 or 3. But the hexokinase 4, which is actually called glucokinase, it is having less affinity because it is called high Km enzyme. So glucokinase can only phosphorylate glucose if the concentration of the substrate is high enough. Its Km for glucose is actually 100 times higher than that of the hexokinase 1, 2, 3. So it will only activate it and will able to phosphorylate the glucose if the glucose is having concentration almost 100 times higher than detected by these three enzymes. Okay. So I hope this is clear, and therefore it will not allosterically inhibited by its product, that is glucose 6 phosphate. Then we have discussed zymogens. Zymogens are inactivated forms of, or the precursor enzymes, which are maintained or kept in such a form so that they can only activated in response to the substrate whenever we take a proper diet, so that they can be activated. Okay, for example, trypsinogen, prolastase, chymotrypsinogen, and procarboxypeptidase all are examples of zymogen. So they are converted, first of all, the trypsinogen is getting converted into trypsin with the help of enteropeptidase, and then the trypsin is responsible for conversion of all the zymogens, prolastase, chymotrypsin, and procarboxypeptidase into their active forms. And we have also discussed one mechanism of activation of chymotrypsinogen into the active chymotrypsin. This we have discussed. Now, what we are going to discuss rest in today's class, the three important things more. One is called ribozyme, one called DNA enzymes, and actually DNA enzymes are actually because DNA is having deoxyribose sugar, therefore deoxyribozymes. And the third is if the antibodies are working like enzymes, then these are called abenzymes. Okay, so these three things are also we are going to discuss in this class, and with this we will actually also have because enzymes are very sensitive for the pH, temperature, and all these things, substrate concentration. So therefore, we are also need to discuss this in one class. But from tomorrow onwards, we are going to study enzyme kinetics. And actually, the enzyme kinetics involves the Michaelis-Menten equation, MM equation, plot, that is a kind of hyperbolic plot. Okay, so we will discuss all these things from tomorrow onwards. So let me quickly finish this rest text which we are left with. Okay, if you are having any query, whatever we have discussed till here, you can ask me. I'm giving you 2-minute break so that you can ask your queries. I hope you're able to absorb at least something in today's class. So there is a question, low Km and high Km point. Okay, I will repeat. Anything else? So see, actually, I need our diagram here. So, okay, I'm coming to other query, but actually I need to explain this one first. Okay, are you able to see my Chrome page? Oh my God, what is this? So actually I need a diagram to explain this in more detail. So this is Michaelis-Menten curve actually. Okay, so we will derive this curve, but for learning actually we all know something about this actually. Okay, so first of all, this is the V maximum. So maximum velocity which an enzyme can achieve, and actually this is the substrate concentration. So there are two axes. One is x-axis where you will represent the substrate concentration in millimolar. One is the velocity. So you will draw the velocity here. So first of all, this is for an enzyme which will follow the Michaelis-Menten equation. So most of the enzymes, including the DNA enzymes, will follow this equation actually, this curve actually. Okay, so if you find some point where this velocity is actually half V maximum, so you will draw a line like this, and then you will draw a perpendicular line. So the point you will get over this substrate concentration line is called Km value, or the Michaelis-Menten constant. What is this constant telling about? So this is telling about the substrate concentration which is required to achieve the half V maximum rate of that particular reaction. Now, we were discussing two important things in isoenzyme section. Okay, let me move to that particular page. So, okay. So we were discussing. Now, suppose there are two kind of isoenzymes. One is called, in one class, we can put all the three. So suppose we have put it 1, 2, and 3 in a class that is called low Km, right? Actually, I'm very writing, I will write very roughly because it is hard to write using this touch screen. Okay, so this is clear. They are having low Km, whereas the fourth one is having a high Km. Okay, so you are not able to understand this. So these are called low Km. So again, I'm moving you to the page. So see here. So suppose an enzyme which is having a low Km, which can be either 1, 2, or third one. Suppose it is having Km value this, and these are called low Km. Okay, so these may include 1, 2, and 3, or you can either say A, B, and C. So these three hexokinases are actually having low Km. So it means if there is less concentration of substrate or glucose, so these are able to sense that particular glucose concentration, substrate concentration, and they will achieve half V maximum. Okay, so this is half V max. But there is an enzyme which is having high Km value. So suppose this is the fourth one, hexokinase 4 is having Km value this. So it will not sense the glucose until and unless there is high concentration of the substrate itself, so that it will not catalyze a reaction and therefore cannot be inhibited by the product of that particular reaction, because the first step catalyzed by the glucokinase or hexokinase is the rate limiting step in glycolysis. Therefore, the rate limiting step will be inhibited by the product of that particular reaction. So what is the reaction product? Obviously, the glucose will be converted into glucose 6-phosphate using this hexokinase. So glucose 6-phosphate will be formed, and this glucose 6-phosphate which is forming will going back and will inhibit this particular enzyme which is catalyzing the reaction. So it will stop this particular reaction. So because they are having less Km value, so they will be inhibited easily, whereas the glucokinase 4, which is also called glucokinase, hexokinase 4 is called glucokinase, it will not be inhibited by the less concentration. Therefore, it will helpful in liver, where the lots of glucose is going to convert into the glycogen. Okay, fine. Okay, so we are moving back, but there are two more queries. Let me look into the queries, what they are asking. So, fine. If you're able to get it, then great. Okay, now we are having. So we have discussed zymogens. We were discussing ribozymes. So obviously, ribozymes. If the RNA is acting like an enzyme or a catalyst, so then these are called ribozymes. So first ribozyme, the term was introduced actually by the Kelly Kruger, who called them RNA enzyme, not the ribozyme. Actually, ribozyme came later, but the firstly introduced by Kelly Kruger as an RNA molecule that is able to catalyze a chemical reaction. Two type of possibilities. One is either they are present naturally or they are synthetic, means the human made. But actually, about the DNA enzymes, they are mostly synthetic. They are very less. They are very rare naturally, but they can be synthesized in the laboratory. I'm talking about DNA enzymes. Okay, so ribozymes are of two types, either natural or synthetic, and they are well known. Before the discovery of these enzymes, scientists think that the proteins are only known by catalysts. So they think like the proteins can only catalyze the reaction. But finally, in 1967, there is a theory, there is a proposal that either these RNA molecules can also act like a catalyst. If you remember Carl Woos, actually we have discussed in our earlier classes, where I told you this Carl Woos is a man who gave three kingdom classification. Okay, so three domain system of life, so based upon two type of sequences. One is called as 16S rRNA sequences, and other is called as and other is called 18S rRNA sequences. So based upon these two type of sequences, these are called signature sequences. Remember something? So based upon these signature sequences and your ribosomal RNA, this is ribosomal rRNA. So he was able to divide all the organisms into three domains: bacteria, that is eubacteria actually, archaea, and eukaryotes. So obviously the prokaryotes are having 16S rRNA sequences, whereas the eukaryotes are having 18S rRNA. Also, these 16S RNA sequences are present in our organelles. So all the eukaryotes are having mitochondria and chloroplasts. I'm talking about plants actually, for chloroplasts. So they are having these signature sequences. Along with Carl Woos, 1967, Francis Crick, DNA model, all of you know, and one more scientist, Orgel, are able to suggest that RNA could act like a catalyst and it can catalyze the chemical reactions based upon finding that it can form the complex secondary structures. So they have just seen that the RNA can make some complex secondary structures, and they give a suggestion that it can work like it can catalyze some of the reactions. There are no scientific proofs at that time, but later two scientists who discovered actually them are in 1980s. So first is Thomas R. Sage, who discovered from this ciliated protozoan Tetrahymena thermophila, and the second scientist is Sidney Altman, who actually working over RNase P. So the two scientists, one is Thomas R. Sage and Sidney Altman, in 1980s give the first scientific proof of RNA enzymes or ribozymes. So these two actually they discovered separately. One is Tetrahymena thermophila ribozyme and second is RNase P. So these are found in RNA transcription and RNA component of RNase P involved in processing of. So what is the function? So RNase P is actually responsible for processing of pre-tRNA when the tRNA is or the transfer RNA. What is the function of tRNA? tRNA are responsible for transferring the amino acids when there is translational or the protein synthesis is taking place in our cells. So first of all, you need to make the tRNA. So pre-tRNA is processed with the help of this RNase P. So they are also giving their, not only showing that they are responsible for some of the catalytic activities. Therefore, they are called ribozymes. Then after 1989, 9 years later, these two scientists got the Nobel Prize, okay, chemistry, for their discovery, because this discovery was very significant in the biochemistry. Ribozymes often require divalent metal ions, and the second thing is these are called metal because these are showing metal ions for their catalysis. Therefore, generally they need divalent metal ions. For example, magnesium ions, two positive ions. So something is also written. Beside these, there are others are also reported later. One is RNase P. You all know group 1, 2 introns, hairpin, hammer-headed ribozyme. Okay, so one is from hepatitis D virus and Tetrahymena thermophila ribozyme, which was the first one actually. So these are all the examples of these RNA enzymes. Now the functions are also given here. Many natural ribozymes catalyze either their own cleavage. Okay, so this is possible that they are going to cleave themselves or they're going to cleave other RNA species. For example, RNase P is catalyzing the cleavage of tRNA. So they also have been found to catalyze the formation of peptide bond. Not only they're going to cleave themselves or some other targeted RNA species, they're also involved in the formation of peptide bond when actually this translation is happening. So the ribosomes are catalyzing the synthesis of proteins. And at that time, these two, one is called 23S, a direct question of CSIR. So this has been asked. So in case of prokaryotes, this 23S rRNA, and then because it is present in 70S ribosomes in prokaryotes, and 80S ribosomes are present in eukaryotes. So 28S rRNA is working like peptidyl transferases. Okay, so they will catalyze the transfer of or the formation of these peptide bonds between the amino acids, so that the amino acid chain can be and encoded, or you can say can be maintained. So they continuously add more amino acids. So to lengthening, for the lengthening of this amino acid chain, so that the complete protein can be synthesized. And then there is another type of which are responsible for cleavage of phosphodiester bond. They are ribonuclease P, RNase P, or this group 1 and 2 introns. Okay. This is not significant. Then we have something else remaining. Okay, so here Okay, so if the RNA work like an enzyme, then it is called ribozyme. If the DNA is working like an enzyme, then it's called DNAzyme. Or because DNA is having deoxyribose sugar, therefore it can also said like deoxyribozyme. So ribozyme, you know, and if there is deoxyribose sugar, the deoxyribozyme. Some other names are also given, that, for example, catalytic DNA, RNA enzymes, or DNAzymes simply. So these are the different names for these DNA enzymes. When DNA is working like a, okay, this we know because they are working like genetic material, they are very stable, whereas the RNA is unstable actually, act as a long term repository of genetic information because they are stable. They can accumulate the genetic information for prolonged periods of times. Therefore, this application we know about DNA. But it was a surprise when we are able to know some catalytic roles of these DNA. So, nevertheless, many experiments have been showing that the DNA can act like a catalyst. So later scientists also find this, that some of the DNA molecules can also work like catalysts. Let us look into more details of these one actually. So actually these DNA enzymes are less known. And second thing is they are not present naturally. Mostly they are synthesized, I mean they are synthetic. While deoxyribozymes are unknown in nature, synthetic catalytic DNA molecules have been identified in vitro selection methods. In some experiments, we have discovered that some of the synthetic catalytic DNA molecules can be prepared. In vitro selection of an experimental process by which random sequences. Okay, so what is the in vitro selection method? In vitro selection method is an experimental process जिसमें हम कुछ random sequences को select कर लेते हैं, in which we select some of the random sequences and we are able to identify some of the major applications of those random sequences, such as those of the DNA and RNA nucleotides. Okay, so are examined in parallel. So because we are selecting some sequences of nucleotides, so we are in parallel we are taking many sequences at one time, not one, to identify those particular sequences which are having our desired function. For example, if we are looking for some DNA which is having some catalytic function, or in some binding or some catalysis. So we only look for that particular sequence out of thousands of sequences we are having in our hand. So this is called in vitro selection method. So using this technique, we are able to identify some of the DNA. These are mostly single-stranded DNA because double-stranded DNA is very, very stable and it cannot catalyze the reaction. So natural duplex DNA molecules are unlikely to catalytic because double-stranded DNA is already very stable, therefore it cannot catalyze the reaction. Because the DNA duplex is structurally a regular structure and the enzymatic activity requires the irregular tertiary conformation. You need to know that whenever there is an enzyme or protein, it should be able to change its conformation so that it can catalyze a reaction. But you know, double-stranded DNA is very stable and it is having some regular structure. Therefore, it cannot work like a catalyst. So mostly the DNA which is acting like a catalyst or an enzyme should be a single-stranded DNA molecule. So this you should remember. That can bind to substrate for catalysis. So the first report about this DNA as an enzyme was published in 1994. So I think most of you are not born at that time. Okay, so December 1994, the first report came in our knowledge when DNA can work like an enzyme. These two scientists, Brecker and Joyce, are giving this particular publication. Since the initial reports of RNA cleaving deoxyribose zymes, many other catalytic activities of DNA. First of all, we are interested in knowing the catalytic activities of DNA, but with these catalytic activities, we are able to report some of the other activities also. For example, in ligation, so it is able to ligate the DNA and RNA species, and also they are also needing some divalent metal ions similarly as are required by the RNA enzymes. So they need some divalent metal ions for catalysis. An unusual DNA catalyzed photochemical reaction, you are familiar with this, photoreversion of thymidine dimers. Yes, so what will happen in presence of UV? Why we call UV is harmful? Because in DNA, it will lead to the formation of thymidine dimers. So reversion. These dimers should be reverted back to the normal condition. So we need some DNA catalyzed reactions. Okay, so DNA enzymes can revert this back. DNA enzyme-based molecular catalysts generally dependent on the metal ion cofactors for catalysis. This we have discussed. It also has been reported that DNA enzyme-mediated catalysis follows the Michaelis-Menten kinetics. So this can be a question. Okay. Mostly, most of the enzymes, whether these are DNA enzymes or some simple enzymes, actually the allosteric enzymes will not follow the Michaelis-Menten equation or kinetics, but mostly most other enzymes which are having only single active site for binding of their substrate, they generally follow this equation or kinetics. Okay, the last point we are going to discuss in this class is Abzymes. What is Abzymes? As you can see, there are two words, ab and there is zymes. What is ab actually? If you remember ab, so we generally write antigen like AG and antibody like AB. These are very similar. Okay, so they can bind together. They are very specific binding. So they can bind to make a complex, antigen antibody complex or immunocomplexes. So these antibodies are having some nature. So this is a direct question. So they are glycoproteins. They are not only proteins, they are glycoproteins, because they are having proteins plus carbohydrates. Some carbohydrate moieties or sugar moieties are added over Fc fragment over these antibodies, so that they are glycoproteins. These are also called immunoglobulins because they are able to mount an immune response. So whenever there is an infection or antigen, for example, some virus, I'm not talking about coronavirus. So some virus or some antigen or bacteria, if infect us, so it will lead to the synthesis of antibodies, specific antibodies, and these antibodies are able to neutralize that particular antigen, so that we can recover. Okay, that bind with the specificity, high specificity to these antigens. An antibody with catalytic activity is called Abzymes. So suppose this is the one function when this antigen can be neutralized by antibody, but what can be the other possibility that these antibodies which are catalyzing these immune response reactions are having some catalytic role. So catalytic role in they are able to catalyze some of the reactions, as we have seen in case of so catalytic antibodies. So some examples are given in this. Okay, so I will discuss. One is actually multiple sclerosis. Okay, so what will happen in multiple sclerosis? It is a kind of autoimmune disease. So in this case, what will happen if someone is having multiple sclerosis? So specific antibodies will be formed, and these antibodies will start degrading the myelin sheath. You know myelin sheath, which is insulating your neurons. So if the myelin sheath is degraded, your insulation is gone. If these nerves are not insulated, you and the foremost thing you should be aware of, these myelinated neurons are having more higher rate of nerve transmission when compared to non-myelinated neurons. You should know this. Actually, this is given in 11th, 12th biology. So suppose if the myelin sheath is lost, so what will happen? It will a gradual destruction of the myelin sheath surrounding your neurons in this autoimmune disease throughout the brain and spinal cord. So what will happen actually? So this myelin sheath is having some protein. So this is called myelin basic protein, which is covering your neuron, which is insulating your neuron. So these antibodies are degrading this particular protein, myelin basic protein, so that these are removing the myelin sheath from the neurons. Okay. So actually these antibodies are having protease activity because they are degrading this protein. They are called MBP, myelin basic protein destructive proteases. So therefore, this was the reported function of these antibodies, where they are degrading some protein, so they are working like proteases. Therefore, they are having some catalytic activity. Okay. So back in 1969, It was William Jenks who proposed that an antigen that would induce the formation of antibodies with catalytic activity if antigen resembles a transition state of an enzyme. So we know the antigen can bind with antibody, and actually this antigen antibody complex is very much analogous to the transition state of an enzyme. When an enzyme is bound to the substrate, so it will form some enzyme substrate complex. So you can compare the enzyme substrate complex with this antigen antibody complex. Later, and another scientist, Richard Lerner and Peter Schulz, verify this hypothesis, and they said this is very much correct, and this can happen actually. And this first verified hypothesis was verified in 1986. Means you can think of there are so many years gap between this 1969 and this was verified in 1986. They independently reported that the two antibodies can catalyze the hydrolysis of either aryl ester or carbonates, respectively. So first of all, Richard Lerner tell about that they can hydrolyze the aryl esters. Then the Peter Schulz in 1986 reported that they can also hydrolyze the carbonates, some of the carbonates. Therefore, they are having some catalytic roles. They use transition state analogs as immunogens. Now, one more important thing: what is immunogen? What is antigen? So, from the first chapter of QA, you will get this. Actually, immunogen is any substance which is able to mount a immune response. So, if our immune system binds some immunogen, so our immune system starts secreting the antibodies. Okay, this is called an immune response. But antigen is any molecule which will able to bind to the, okay, so antigen is any molecule which is specifically able to bind to any antibody. Okay, so antigen and immunogen, what is the relationship between the two? So you need to note this line: all immunogens are antigens, all immunogens are antigens, but it is not true in the reverse way, because all antigens not necessarily are immunogens, because it can happen that some antigen are not able to mount an immune response. Therefore, all antigens cannot be immunogens. But remember, all immunogens are antigens, because all immunogens are able to bind to the antibodies, and therefore not only antibodies, they are also able to bind to the B and T cells which are having some antibodies bound to them. Therefore, they are able to mount an immune response. So you need to remember, all immunogens are antigens, but reverse is not true always. So that mimic an unstable tetrahedral intermediate of a reaction. The hydrolysis of aryl ester and carbonate, which was reported in 1986, a well-known reaction involving the formation of breakdown of the unstable tetrahedral intermediate. So this can be deemed to be closely related to the transition state of a reaction. If you remember enzyme substrate complex, so the antibody can also form a complex with the substrate, so that it is very analogous to the transition state of a reaction. A variety of catalytic antibodies have been generated after that, and catalyze a wide variety of reactions using these haptenic transitions states analogous to the immunogens. Okay, so there is another molecule, haptane, also, which will enhance these immune responses. Now, this Abzymes have a great potential because they are able to catalyze the reaction, and they are having very applications. Therefore, they are very important, great potential in the pharmaceutical industry. How we can use them? Abzymes have been implicated for the use of detoxification of the cocaine. So, if you want to remove the toxicity of the cocaine, so suppose some addicts who generally use cocaine as a drug, so this can be used, antibodies can be used to detoxify the cocaine. So this can be potent pharmaceutical application in the industry. These antibodies have been generated that cleave the cocaine molecule at a specific bond, thereby eliminating the toxic effect of the drug. So perhaps the most exciting application of the Abzyme technology is the specific targeting of the cancer cells for destruction. Not only the cocaine detoxification or the removal of the toxic effect of the drug, they can also be used, they are having very potent applications in the cancer treatment. Okay, so they can target some of the specific cancer cells, and they can able to remove those cells out of the body, okay, by catalyzing or proteolytic cleavage of some of the proteins over the surface of these cells. Okay, so they are having application in cancer biology. They are having application in detoxification of cocaine. They are actually they are also shown to involve in multiple sclerosis, where they are working like proteases. So therefore, as a conclusion, we can say these antibodies can also work like catalysts or enzymes. Therefore, they are called Abzymes. So today we have learned many things. We have discussed that there are some specific enzymes, for example, aspartate proteases, which include HIV protease, which include pepsin, renin. Then there is another family, serine proteases, which include different. So they include trypsin, chymotrypsin, elastase, thrombin, plasmin, calcarein, acrosomal and lysosomal proteases. All are these serine proteases. So all the serine proteases are having some catalytic triad. Then we also have discussed that an enzyme can work using one or more catalytic strategies, either covalent catalysis or acid-base catalysis, or for example, metal ion catalysis. So this also we have seen. And there is one more important thing that is called proximity and orientation hypothesis. If you want to know what is more important, so obviously the orientation effect is more important, because the proximities do not show great effect over the enzyme catalysis. It is always the orientation. If an enzyme is able to orient the substrate in such a way that they can interact physically with each other, so that they can be converted more efficiently into the products, therefore they can increase the rate of reaction more efficiently. Fine. So with this, actually, I'm concluding today's class. If you're having some problem, you can ask. I'm unmuting you all. And tomorrow onwards, we will look into the Michaelis-Menten kinetics and other aspects of enzyme kinetics. So that is very important. So I was waiting for the students to take the class maximally, so that we can